Hey, Bio 2 students, Mr. Yance again. Hope things are going well. We're going to continue on with notes and kind of go over a few things with degrees of dominance and some things like that. Hopefully the Punnett Square quiz went well and you understand a little bit more about how those work. So I just want to kind of explain that a little bit more. So unfortunately, a lot of genetics is not just simple. It's dominant and recessive. Okay, I wish all traits were just dominant or recessive. It doesn't work that way. A lot of them you can predict that way, and that's what Mendel was trying to get at. The relationship between genotype and phenotype is a little more complex than maybe what Mendel explained in his pea plant experiments. He was very fortunate. He picked a plant that did not show any of these strange characteristics, which I'm going to talk about today. They had dominant genes and recessive genes, and most traits are that way, but it's rarely that simple. Many traits, or he called them characters or factors, we call them alleles now. There might be multiple alleles. I think a great example would be like blood typing. You got A, you got B, you got AB, you got O. Eye color, you got green, blue, brown, hazel, and here's the thing I want to get across to you. Just because I'm going to show you some variations and some different things with this, it is not going to violate any of his laws. The law of segregation, law of independent assortment, they are still at work. They always will be at work. That's how meiosis works. They're going to come together at the equator and separate, and we're going to get a sorting of traits. But it just doesn't explain all of what goes on. So we're not going to violate any laws or rules here. It's just a simple case of there are some exceptions to the rule of dominance and recessiveness. And that's why dominance and recessiveness will never be a law, because there are these exceptions to that rule. Okay? So what we've been talking mostly about in most Punnett squares, and we did something like this, is complete dominance, right? You have a dominant trait if it gets paired up with recessive, like the example up here. I got yellow seed is dominant over green seed. That basically is pretty straightforward with genetics. However, we don't know when it's a heterozygous which one a person is. We did some Punnett squares like that where we didn't know the one parent, and we had to look at the offspring to see if he was homozygous or heterozygous or she or whatever. It doesn't mean, one's more yellow than the other or one's taller than the other. They exhibit the same phenotype. Okay, so that's a little bit of a problem with complete dominance sometimes, is with those dominant traits. And it's really important when we're breeding dogs or cats or cattle or horses or whatever, and I want a particular look, I want to make sure I have homozygous traits. I don't want these heterozygous throwing me an animal that's going to look odd. It's not going to be able to sell. So it is important to know that, and we'll kind of circle back to that in a second. Incomplete dominance, let me put both these up there. Incomplete and codominance, same Punnett square, but different look in the organism. Okay, so let me try to explain this so it's not confusing. With incomplete, I am going to get a blend. So let's say I have a red flower and a white flower, and I bring them together and I get a pink one. They blend. Okay, codominance is where I got two dominant genes, just like I do with incomplete, but instead of blending, they're actually both going to show up. So like in horses, we have a thing called roan where red's dominant and white dominant, but you don't get a pink horse. That would be interesting. You get a roan where the red and white hairs grow side by side, and you get a speckling. Okay, so you set up the Punnett square the same way, and you use the prime thing like I showed you. But you got to understand they are different. Blood typing is a great example of this, okay? And I'm going to put this one up there because it goes with that. It also, blood typing is crazy because you got multiple things at play here. Okay, you have the codominance thing going on where I get an A and a B, so they both show up. So you got, if you're an A-B person that has that blood type, you have both of those genes show up. But it's also called a multiple allele, where we have an A, a B, and an O. And then it even gets crazier because then you get all this type of crazy thing going on. You might be homozygous, you might be heterozygous, you might be AB, you might be O. You got 4 different possibilities here. Then you throw the RH factor in there. That makes it even crazier yet. So this is where we had a lot of problems with blood transfusions when we first started giving blood to people because we didn't understand the genetics. And a lot of people died because I have A blood and we didn't know any better. We thought blood was blood. I have type A blood and someone gives me type B blood. That's not good, right? Because you can see it produces these carbohydrates on the surface, and you can't give, B blood to an A person, A blood to a B person. You have to be super careful what blood typing you use. Now, you can give an O to anybody because it doesn't have anything on the surface, but you have to be careful mixing blood. So you can see the importance of genetics. If we go back a slide, I kind of skipped over that real quick. This is that incomplete thing. Red, white, and I get a pink, something in between. So, there's lots of things going on with genetics, or it's complex. It's not just straightforward. So let's just dive right into some more craziness. Then we got this condition called pleiotropy. Pleiotropy is a property of a gene that causes multiple phenotypic effects. So you have a sickle cell anemia, a person who has that disease, that's one defective single gene. But unfortunately, that gene expresses multiple phenotypic effects. So you can't just say this gene produces this phenotype. That gene may produce 3 or 4 phenotypes. Okay, so it is sometimes hard to diagnose diseases and talk about here's the symptoms you're going to see. It just depends on what gene's defective or mutated. And then the other thing that you got to throw in there is, where's that mutated gene, what part of it is affected. So what phenotype is it going to affect? So there might be multiple things. So that's what pleiotropy means. Epistasis, this is going to get more complex as we go, sorry. That's where we got a gene at one location affects the gene at another locus, okay? So I love this example because I think we need to understand skin color, because we get so hung up on white people, black people, brown people, whatever. All these different races of people we have. And unfortunately, our culture is still very racist. There's a lot of that still goes on. And it's really crazy, not just from the moral aspect of it, but it's even crazier yet when you're talking about the genetics. Because watch this. This is kind of interesting, and I think something we need to understand. People that have a gene for heavy pigmentation, that's a separate gene that produces that expresses the trait. So heavy skin pigmentation is affected by another gene in another location. Okay, so what happens is the gene for that pigment, you might have a dark skin pigmentation, but you may be very light in tint coloration because you have another gene that's affecting that. Okay, that's called epistasis. In other words, gene over here is affecting a gene over here. Okay, now it even gets crazier when we throw this next thing in. Okay, like albinos. Okay, here's albino. They should have this pigmentation here. These two are sisters, but this one got the defective albino gene, and that gives her white hair and really light colored eyes and things like that. So see, that's the thing you got to understand is crazy genetics. Okay, and then you throw this one in there, and it even gets crazier yet. We call this polygenic inheritance, where 2 or more genes have an additive effect on a single character. So this is what I was talking about, how people get so hung up on skin color. But look at this. Most of us are in the bell curve. Most of the population are skin tones that are light brown, dark brown, or malto, which is a blend. Very few people are way to the right, which is pure black, or way to the left, which is pure white, which would be an albino. Look at the numbers. It's crazy on the proportion of the population. Most of us fall in here. So that's why I just think it's insane that we get so hung up on race and skin color and things like that, because you know what? We're all a blend, and you can see that we all have this polygenic inheritance. And does it really matter what our ancestors, what kind of pigmentation their skin have? Humans are humans. And I think we need somewhere, someday in our society, get beyond this racism stuff, because it shouldn't be there. So you look at the genetics. You can see what I mean. All right. Okay. So that's kind of what we're talking about. So I just kind of want to introduce this one. I'm kind of a little bit ahead of myself because I haven't even got to this in my traditional class yet. So many humans do follow some patterns of inheritance. There are a lot of things that are predictable, even though it's complex, and you can have this epistasis and polygenic and all these different things going on. You still can do pedigrees. And pedigrees is just a family tree that describes a relationship between parents and children, so we can track genes in a population. It's really good to know your ancestry. And I'm not just talking about Ancestry.com where you see, oh, I'm from Europe or whatever. It's really important that we're able to track traits using pedigrees. All right. So I'm going to get into this. I'm going to put an assignment on Google Classroom coming up here. So I'll explain this more in the next video. And we will kind of break this down so it makes sense. All right. Kind of a shorter video today, but I hope that makes sense. We're all caught up now. So, hey, have a great day, and we'll see you later. See you. Bye.